Thanks a lot, Christine, for your kind introduction, and thanks a lot for the honor of inviting me to give the bidder lecture and tell you a little bit about the work we've been doing in a wider context over recent years of the Intergovernmental Panel on Climate Change. In a way, if you work for IPCC, it comes with a virus, and you kind of start a career, and this career has now taken me into a leading role, becoming co-chair developed country of one of the working groups. And this 3 working groups in the IPCC, and this working group is looking at assessing research on impacts, adaptation, and vulnerability. And we are doing this, and I'm co-chair for the 6th assessment cycle. At the same time, looking at impacts, going from impacts in the marine environment, like the bleaching of coral reefs, or the melting of ice and what it means for natural systems, or droughts and floods. This is all important information, and that has been guiding policy decisions. And it has been guiding and is still guiding ambition in adaptation and mitigation measures that are so much needed to set up a more sustainable world. So influencing policy decisions, that's what the IPCC work is about, and it has been important in the context of the Paris Agreement. And down at the bottom figure here, you see as in the process at the last approval session of the outline of the 6th assessment report in Montreal last year. Now, IPCC products have made an impact over time. The first assessment report, and we actually are just celebrating the 30-year anniversary. The first assessment report led to the foundation of the UNFCCC, the Climate Convention of the United Nations. The second had input into the Kyoto Protocol. The third then focused on impacts of climate change, need for adaptation, and adaptation in an IPCC context usually means human adaptation measures. The fourth report emphasized the decision on the 2-degree limit, and then the 5th assessment report that I had the honor to be involved in as a coordinating lead author on one of the oceans chapters in Working Group 2 provided input to the Paris Agreement and emphasized the need for ambitious climate mitigation, leading us hopefully to a 1.5-degree world and not beyond. These products are important also because just recent insight has emphasized the need to also consider the Sustainable Development Goals of the United Nations in this context, and all of these goals should be reached by the year 2030. And it is very clear that reaching climate targets is linked to reaching those Sustainable Development Goals, which tells you that we are actually living in an era of high urgency of taking action on climate change. Let me focus on one of the SDGs, SDG 14, Life Below Water, because that is my specialty. I've been working in the marine environment for years, and I will talk you through how the IPCC works and using examples from the last assessment report illustrating how we got to the respective conclusions. So according to emission scenarios, the oceans are warming, and we are distinguishing different trajectory according to emission scenarios, the so-called representative concentration pathways, and the radiation load for the Earth. And what you see here is a combination of observations, then the futures that we are moving into towards the end of century, depending on whether we are going into ambitious climate mitigation or whether we let go and have business as usual scenarios. We are currently living in a period of committed climate change where it doesn't make so much of a difference which scenarios we are using, but we are now making the decisions on our climate future, and this disconnect between what we have to decide now and what is going to happen later in the century is certainly part of the problem and part of the current inertia. At the same time, the oceans are acidifying. You see here the pH depicted for historical time. We have already seen a decline by 0.1 pH units, And depending on emission scenario, we either go into a balancing of the acidification or into a business as usual and unabated acidification trend for the oceans. And then losing oxygen, and here's where the word underestimated plays a role, because if we just look at the average numbers and the percent changes expected, they seem very low, and the changes may seem very low on historical timescales and also in the projected timescales. But what I'm learning from the climate physicists at this point is that obviously the degree of oxygen loss to the ocean or in the ocean has been underestimated, and especially that is the case for the low oxygen areas in the oceans, as there are the mid-water oxygen minimum layers or the coastal oxygen deficient zones, so-called dead zones even, which are expanding. So this is a serious problem and will keep us busy for the future to look at. How to put climate findings into context. That is something, how does it influence decisions? How do we get there? What kind of information is needed to influence policymakers to take action? That is certainly an important question. And there is the Climate Convention of the United Nations that makes their respective claims. We should prevent dangerous anthropogenic interference with the climate system. This means we should allow ecosystems to adapt naturally. We all know that organisms adapt, but not ecosystems. And then we ensure that food production is not threatened, and we should enable economic development to proceed in a sustainable manner. So the question really is, which degree of climate change is dangerous? How do we capture adaptation, human and evolutionary adaptation in that context? And how do we link climate to economic development? That's certainly a point I cannot really go into during this lecture. The reports that the IPCC prepared are structured into chapters focusing on themes, high-level themes, like, for example, the oceans theme during the last assessment report. And these are assessments. And this is a term that if you focus on your research, on your basic science activities, you would not normally use because you're used to the term review. But an assessment is not just a review. It is intended to objectively summarize the state of knowledge in a balanced way, including an assessment of confidence and uncertainty. It is to be policy relevant. So basic science information, although often found in chapters in a certain context, is not really the core focus of the IPCC. It is to be policy relevant, therefore it need not comprehensively cover all of the literature in the field. We have to be selective there, especially since that literature has been exploding over recent years. An assessment is a consensus document produced by the author team. It's not a collection of individual contributions. Assessment statements should be agreed as a group based on expert judgment. And also there is a tightening of the language that we use. There should be precise, calibrated language as an essential element, especially in the summary for policymakers that will be negotiated in a one-week plenary, word by word, line by line, with the 195 government delegations that are members of the IPCC. There's another aspect to be emphasized, which is kind of different to the normal world, but we could think about to what extent this might be useful to adopt actually, and that is the principle of detection and attribution. So the detection of change in a system is defined as a process of demonstrating in this context that climate or a system affected by climate has changed in some defined statistical sense. This concerns the physical climate sciences, Working Group 1, and it concerns Working Group 2 because the system affected by science, that's sort of one step further down the line. So when we then say, okay, there is a change and we think it's affected by climate, then the attribution to climate is an important step as well. So is defined as a process of evaluating the relative contributions of multiple causal factors, at least some of them have to do with climate change, to a change or event with an assignment of confidence. And then certainly a permanent discussion between Working Group 1 and Working Group 2 is to what extent we do attribution just to climate change, whatever the cause of that climate change is. This is something we've been doing in the Working Group 2 because we don't have a direct handle on what is the anthropogenic influence, except maybe for ocean acidification. Or, and this should be the future, we should try and disentangle whether there are natural climate changes in a region that are important, or whether this is the anthropogenic influence that plays a role. So all of these are important steps, systematic steps that not necessarily you would take into account in your experimental research. But just to illustrate kind of the level of evidence, the multiple lines of evidence that are being drawn together in a conclusion concerning climate change and climate change impacts, I will talk about a little bit about the coral reefs that were identified as a vulnerable ecosystem in AR5 with a high level of confidence, and these reefs are under various pressures. Distinguishing these various pressures from the impact of climate change is important, but certainly the prominent influence is by temperature, where a healthy coral community can go into and is increasingly going into a bleached community, losing color because they lose their endosymbionts. If this process is continuing for several weeks, then these corals would die because they lose their energy provision system, and the coral would turn into something like an underwater desert. And these observations have already been made in an 0.8 degree warmer world, and we have currently already surpassed the 1 degree global average temperature increase compared to pre-industrial temperatures. Now, if we look at what has been observed, then there has already, since the 80s, been a loss of the coral cover in this system, and this is the Great Barrier Reef, by 50%. But it's due to various drivers. There are the crowns of thorn starfish, predatory starfish working on these corals, and there are high-intensity cyclones that have caused destruction, but there has already been a contribution by bleaching up to 2012. Now the bleaching is exacerbated. And you see here with the recent El Nino event, an increase of bleach surface area for the Great Barrier Reef. And if we look at the projections that have been made, then there we have a high risk of losing up to 90% of healthy coral reefs and their services to humankind, even in a 1.5 degree warmer world that we will have reached by the 2030s and 40s. So drawing multiple lines of evidence through system understanding is important, and just focus on the right line here. But the kind of evidence that is being put together is that the knowledge of warming, of ocean warming, has been linked to greenhouse gas forcing. That is the underlying information coming from Working Group 1. And then over the past 490 million years, coral reef die-off has coincided with increases in CO2, methane, and or warm temperatures. So we have a paleo reference, which is very important for the long-term estimation of effects. And then there was a first observation of Mars coral bleaching in 1979. And they have consistently followed after that with increasing frequency following warm sea surface temperature events, for example, caused by El Nino. And now there is where experimental biology comes into play. Laboratory studies show coral bleaches under stresses, such as warm temperatures, salinities. We know there is a synergistic impact of ocean acidifications, and so forth. And substantiated by current observations of repeated losses of tropical coral reefs with warning events, and they are documented in the scientific literature, and meta-analyses are an important instrument to pick up that information with the right kind of hypotheses behind. So experimental biology comes in here. studying the responses, studying the mechanisms, and bringing that information, providing a basis for those conclusions. We can go beyond calls and go across organisms and look at temperature stress and temperature limits as a general principle. And we've done that during the last assessment report. We've looked at heat vulnerabilities in a meta-analysis, considering them in marine species from different environments. And what this slide shows is that consistently for plants and animals in the oceans, these limits are in the low 40s. And actually, this is touching where we actually find the warmest places in the world's oceans. So there are places in lagoon systems and so forth which become warmer and no longer habitable by higher marine life, whereas unicells or bacteria and archaea we shouldn't be so concerned about because they obviously are able to tolerate higher temperatures if we just look at the influence and role of temperature. And an interesting finding for us in that context, trying to understand the system here, was that the complexity of the organisms related to the highest temperature of growth for these organism groups, and the highest complexity organisms, the multicellular eukarya that we are looking at in the SCB, are the most sensitive to thermal extremes. So complexity setting evolutionary limits. These are evolutionary brick walls. There's no way that these organisms would be able to evolve to live at higher temperatures. This is their evolutionary constraint. Another mechanistic study at the time, which was kind of a proof for a hypothesis that we had developed in that context, we saw that there are climate effects in the field for benthic fish species in the North Sea. Under temperature extremes, we see a decrease in abundance. If we relate that to their temperature-dependent performance, in that context its growth, we see that the loss in abundance happens at the same temperature range when there is a decline in growth. And we could, using our NMR facility, explain that by physiological findings. We saw a leveling off in blood flow, so there seemed to be a circulatory constraint setting in, whereas the extreme oxygen deficit indicated by an accumulation of succinate in the liver would come in late. This has several background implications in terms of mechanisms that are being involved in this. Growth includes acclimatization and energy budget trade-offs. Growth studies always take a long time, and they are a pain to do. At the limits of acclimation capacity, obviously, there's a loss of fitness performance capacity measured as reduced growth, causing reduced field abundance. And limited growth relates to limited cardiocirculatory capacity in relation to demand, supporting the hypothesis of oxygen and capacity limited thermal tolerance. So these kinds of fundamental information are important to support the detection and attribution that is so important in these assessment reports. And there are different aspects, high-level observations that are there to consider. There are range shifts of fish and macroalgae happening, changes in community composition, changes in abundance, impacts on large non-fish species, walruses, penguins, and other seabirds, impacts on reef-building corals, changes in fish species riches. These are all examples where during the last assessment report, the conclusion on confidence for detection and attribution was high or at least medium. And here, the experimental work supports the understanding of the system, supports the understanding of their obligatory responses to a climate challenge. So experimental biology can contribute to a mechanism-based cause and effect understanding, helping the detection of impacts and their attribution to possibly even anthropogenic climate change. Most ideally, certainly, through identifying unifying principles of impacts and adaptation across phyla, that would make argumentation at high levels so much easier, and building consensus, drawing together multiple lines of evidence, including beyond experimental. So making connections to other scientific disciplines is a very important part of the process. Now, how do we assess and characterize confidence in those reported findings? And we have a guidance note for lead authors of the assessment reports. We are going to use this also during the 6th assessment cycle, where actually this is laid out, how we do a consistent treatment of uncertainties. What essentially confidence assessment means is that we bring together the level of agreement in the scientific community and the level of evidence seen. So the combination of these go into different levels of confidence, as you see here, from very low to very high. And certainly the best is if there's high agreement in the scientific community, if that ever happens, and whether there is robust evidence underneath. And as I indicated in the beginning, it really needs the author team to discuss and come to grips and come to a consensus on these terms. There could be robust evidence and low agreement. So various combinations of these items are important in then finally coming to a confidence statement which even makes it then into the summary for policymakers. And in the rich diversity of findings reported in the 1,500 pages of the last Working Group 2 assessment report, you see these are some core examples listed on a global map. And the intention certainly is to do better next time, provide a better regional resolution, and provide more examples. So there is an effect on physical systems, the blue color. There's an effect on biological systems and human and managed systems, not to forget. And whether the impact is strong is shown by full or open symbols for the different. And the confidence level is indicated by this bar here. So there are various ocean impacts that have high confidence just listed here, but there are also impacts in terrestrial realm and so forth. So this was a figure that then made it into the summary for policymakers because this is policy relevant information. But let's now go back into some more detail with respect to physiology, the role of experimental biology in that context. If we go through meta-analysis of the literature, one of the main results was that organisms are on the move. And this concerns all organisms virtually, except for this group of non-bony fishes, where the evidence was scarce at the time and not sure what it still is. Looking forward to see the next assessment in the chapters. But all organisms obviously are on the move, are not able to stay behind and fully adapt to the warming trend. The driver here clearly is temperature, and these changes have been observed in an 0.8 degree world, and they move by tens of kilometers per decade, some groups even faster and stronger than that, telling us something about adaptation, about their velocity of their capacity to adapt to the new situation. They are obviously falling behind in that capacity. So what are the physiological underpinnings? How do we explain these observations in animals and across organism domains? Well, for animals, there is a long tradition in the ecological literature to evaluate the lethal temperatures or the City max values close to lethal. And these are the high and going across, and this is for fish, going across latitudes. This shows how the temperature maxima, the lethal limits, go down as we go into the high latitudes, and there are lower limits that also go down as we go in the high temperatures, and this shows the temperature where the fish likes to be. So there seems to be a trend, and this data meta-analysis indicates that there is a narrowing window towards polar areas. You can ask the question, okay, why is that? What are the constraints? Why is that? What is causing that? But there is certainly one aspect to be considered. Ecosystem effects are often due to sublethal constraints on organisms, so just looking at lethal temperatures will not give us the full information. Now, there's one paradigm in evolutionary biology, and that goes across organism domains, and that is the so-called thermal performance curve. You find it in bacteria, you find it in unicellular algae, in plants, and in animals. And this is a paradigm we adopted in terms of coming and from a high-level question, providing a high-level answer, indicating organisms are specialized on limited temperature ranges. These temperature ranges will indicate where they overlap with respect to their temperature range. This is the temperature range of coexistence, and the width of the windows is narrower towards polar areas, and the dynamics of the thermal range indicated here by the dotted lines indicates already at the time there was evidence that showed that these thermal ranges would respond to additional challenges like ocean acidification or hypoxia in the oceans. But this could be the basis of changing species interactions and feedbacks. Interacting drivers of change would also be considered, but they are not yet considered in the modeling, and we will show the importance of modeling in the next step. So having an overview like this would be nice, but from an experimental point of view, we are looking at individual species, we are looking at their genes and how all of this works together, but are we really looking out at the comparative physiologies of interacting species in the field? That is a question that is certainly an open one and should be one addressed in the future. But the question to be asked in general certainly is, can we, and this is together with Sinclair and colleagues just in 2016, can we predict ectotherm responses to climate change using thermal performance curves and body temperature, certainly considering one aspect, which was also important to us, that these performance curves may be different among life stages. And looking at the life stages and what they mean in an ecological context for the species and species fitness is an important concept at ecosystem level. So from an experimental point of view, we need proxies for these overall thermal performance curves, which are kind of fitness curves, but hard to measure. Considering that the thermal performance curve is plastic, a variable between life stages, we think ROSE is a suitable proxy. Others think that aerobic scope for exercise is an excellent one, and actually both of them may work depending on what the species actually does at ecosystem level, and that is an important aspect to consider. Certainly, recent insight emphasizes tests for CT max and aerobic scope for exercise in exhausted or stressed individuals will give relatively poor results in this context. No response to that, okay. Not all thermal windows are the same. Just to emphasize that again, and thermal windows go from eggs, early larvae, narrow, towards the juvenile, having a wide range, growing adults, and then as the species or the individual becomes larger towards the spawning stage, they become thermally sensitive again. So we need to consider an allometry of thermal specialization, and we need to consider developmental constraints on thermal window width. And just to emphasize, as an example, there are findings in aquatic insect larvae that fit into this context as elaborated by Wilco Verberg and his colleagues. So what we need are coherent analyses across species life stages, but the availability of those is rather limited. Just as an example for the allometry of thermal tolerance here in Atlantic cod, an important fisheries species, as you all know, we see that in smaller individuals the thermal range is quite wide, and as the individuals become larger, upper thermal limits go down in respect to their thermal performance curve. And then if we go even further, there's a strong specialization and reduced heat tolerance in larger specimens. And if we think about them accumulating even more weight when they accumulate the eggs and sperm, this is certainly an aspect that needs to be considered as well. But this is not the full story. There is also a dynamic change in these thermal ranges, in the thermal performance curve, depending on where the species or even the population lives. And here's a differentiation between populations of Atlantic cod, Iceland cod, Norwegian coastal cod, and North Sea cod. And you see, in principle, the performance for North Sea cod is better. They have a higher upper thermal limit, and it's lower for the populations further north. So there seems to be a trade-off between where the species live, the temperature variability, and the energetic consequences for the species. And this observations from the lab has been confirmed in the respective field data. We know that the populations are different with respect to their energy demand at different temperatures. So there is something, and this is northern hemisphere latitude, there's something like a cold adaptation a challenge for the northernmost populations. We see that reflected. The higher energy and oxygen demand of the southern populations allows for higher venous PO2 levels than, for example, in the Barents Sea, and that may relate to the higher heat sensitivity of the northernmost population. So these high-level whole organism considerations are important to consider, and just this example, and this comes from a completely different organism, comes from yeast, Saccharomyces cerevisiae, but the principles are the same. We have a thermal performance curve related to growth, and if we look at the temperature limits for individual enzyme systems, we can see that all of these temperatures are beyond the denaturation temperatures are beyond the range where actually the yeast is growing best. So the integration of all of these individual mechanisms into the whole organism is something that should always be behind in experimental approaches and be considered. Otherwise, there may be pitfalls in simplification. And this is true for animals as well. If we look at data in one of our model organisms, the Antarctic eelpout, Paricara brachycephalums, if we look at isolated mitochondria or hepatocytes compared to whole animal changes, we see that these are functional at much higher temperatures than if we look at the whole organism. Heat limits go up as we pick a system from the whole organism. But there's an even narrower window for performance, for steady-state performances like growth. And this happens when the whole animal oxygen demand is lowest. So the energy budget of the organism and what it uses its energy for is an important consideration in these considerations of steady-state performance. So optimum growth occurs at lowest baseline cellular and animal costs. And this led us relatively early to say there must be a systemic to molecular hierarchy of thermal tolerance in animals, the highest functional complexity and integration setting the thermal limits, and the matching supply and demand is an important example. We saw in the eelpout that the circulatory system is relevant, and there are further data by other groups that are emphasizing and pushing along the same lines. It's unclear as of now whether this is a unifying functional principle across domains. Looking at supply and demand might be an interesting hypothesis to follow through. And we think this is captured by the so-called OCLTT concept, the concept on oxygen and capacity limited thermal tolerance. And it pays off to work towards integration, to look at systems in a whole organism context. So towards analyzing integrated systems. Recent works, for example, indicate that mitochondrial limitation may occur at lower maximum temperatures in permeabilized cardiac fibers, and Tony Hickey has been pioneering that field in the comparative world. So a close correlation with whole animal data is certainly needed to address such hypotheses. And this also concerns, and we are joined forces with the group, concerns acclimation studies in mitochondria, which show close responses to the environmental changes that are occurring in those systems. So thermal performance curve shift at the whole animal level, depending on what's happening at the lower organizational level, and there's certainly a trickling back between the different systems. This may be a simple left or right shift of the performance curves, as nicely illustrated in a recent review by Sebacher et al., or it may relate to something different, the so-called counter-gradient variation in growth seen in a North Atlantic silverside species. But there is a commonality. The acclimation concerns about shifting the point when functional limitation occurs towards high temperatures. And getting a grip on the functional limitations, the reason and the mechanisms underpinning functional limitation is an important issue. And these acclimation dynamics can even be seasonal, as we see here an example from a study in one of our beloved worms that we also work with, is how, and you see how the baseline rate of oxygen consumption changes between winter, summer, and spring. But it's common for all of them. They have an exponential nature. There is a clear Q10 effect on oxygen demand and reflect a changing width of the temperature window. And these are putative performance curves set on top to show that this baseline energy demand is then influencing the upper energy demand base or oxygen supply based on that. And mitochondrial acclimation is part of this, and you can see that this is different whether the fish lives in a constant temperature environment like the Antarctic fish, and early findings in fish and bivalves showed that the mitochondrial capacities here are really at the low end of the capacities of mitochondria studied. But then if we go into the northern hemisphere latitude again, where we have high temperature variability, we all of a sudden see that species can double their mitochondrial capacity to live in a variable temperature environment at the low end of the temperature scale. And in Aranicola, compared between White Sea and North Sea populations, actually mitochondrial density differs at the same temperature by a factor of 2.4, meaning they are adapted to different temperature means and variabilities. And that's an aspect that we shouldn't forget when we do our further work. So the complexity of the interacting factors we have been put together in this concept of oxygen and capacity limited thermal tolerance. I've just focused on the relation on the mitochondrial functions and their changes, but certainly energy consumers are important as well. Then as we go into further temperature extremes, there are protection mechanisms like heat shock proteins and metabolic depression mechanisms or shifts to anaerobic metabolism that are important, and all of this occurs before actually we have a damaging effect on macromolecules, and there we even have mechanisms that are involved in their repair. And the thermal performance curve nicely fits into this aerobic window by showing its optimum close to the upper, what we call Peyer's temperature, the upper limit, thermal limit of maximum function, if we want to call it that way. And the thermal performance curve relates to steady state functions like growth, reproduction, development, foraging behaviors, and immune capacity. So fitness supported by routine steady state functions should be at the core of the experimental work done in this context. And this is indicating that these curves may respond to other challenges like hypoxia, CO2, or biotic stresses. Acclimation can be complex. We did a long-term study in the Antarctic eel pout. We saw shifts during warming of aerobic capacity, citric acid cycle, carbohydrate metabolism, lipid metabolism, and some regulatory factors. But there was one finding that I found especially intriguing. If we look at the growth curves, the gene expression response of upregulated and downregulated genes, sort of the mess going on at the gene expression level, was minimum at the temperature of optimum growth, telling that this effort at that machinery is also adjusted to the temperature of where the species performs best. The role of oxygen in thermal tolerance in the terrestrial world is currently challenged and under debate. But there are some findings indicating that there are some oxygen safety margins, that oxygen supply limits the heat tolerance of lizard embryos, or that oxygen concentration affects upper thermal tolerance in a terrestrial vertebrate. But still, the universal applicability of OCLTT as a basic evolutionary framework is still controversial. I think that's my impression. This may be due to less appropriate proxies used or less appropriate methods used, something that we have debated for quite some time. So we would need additional methods, maybe that are useful to show us the subtleties in the changes involved. And certainly at some point, to be relevant at policy level, we should develop some consensus on this. So there are new methods and indications. Actually, in a Drosophila study, we originally thought, oh, well, insects are an exception. But then this study found genotypes more likely to fly under hypoxic stress were also more likely to fly under heat stress. This finding prompts new questions about mechanisms and limits of adaptation to heat stress. And then there is another study showing the influence of omics approaches, in this case metabolomics, where in moth larvae, terrestrial moth larvae, there are hypoxia symptoms in midgut mitochondria, an increased transcription of hypoxia-induced factor 1 alpha, and heat stress caused oxygen limitation, depressed the overall intensity of aerobic metabolism, and they sustained their energy supplies through anaerobic glycolysis. So that would also indicate that these principles still work. They may be modified, they may be adjusted. We need a diversity of approaches to still evaluate whether this is a unifying principle or not. A question I would like to ask in this context, going back to ecosystem level findings, is when we talk about aerobic scope in species exploiting their aerobic scope, how much steady-state aerobic scope is actually needed at ecosystem level? And here I would like to emphasize the usefulness of modeling experiments, because they could test to what extent physiological principles can help us explain field observations. And a finding from this modeling study we did in 2015 was that the upper heat limit obviously was related to a critical metabolic index, as we called it. But essentially what it is, it tells us about the minimum level of routine metabolic scope that the animal needs to have available all the time. And this seems to define the southern distribution limits of Atlantic cod, considering they are living on the shelf, on the eastern and the western side of the Atlantic. And this would actually also explain how seasonal migration of this species occurs. On the western side, there's a migration between 41 degrees north and 37 degrees north, depending on the seasonal temperature regime following this critical metabolic index. Whereas on the eastern side, there is a migration to deeper water, also characterized by this critical metabolic index. So these are barriers felt early by the species based on their physiology. Now, debates about cause and effect are useful if resolved, and if we come to consensus finally. A debate can entertain our own community, But in order to be important to other species, not to other species, but to other disciplines, we need to have consensus messages. And there is another aspect to be considered. Time is short. How do we help the climate debate in times of urgency and high need for policy decisions? In the Working Group 2 report, we have developed a concept addressing risk. And we go across sectors, something you would find from an experimental biologist's point of view unbelievable, how you can compare risk across and assess risk across sectors. But it's a very useful exercise. We are using the so-called burning ember diagrams, where we characterize risks, levels of risk between undetectable, moderate, high, and very high. And we are, and these are the so-called 5 reasons for concerns, and only the left one is the one relevant for us at this point, let's say it that way, because it considers unique and threatened systems like warm water coral reefs. And you see that with different global mean temperatures, we are crossing different risk thresholds. And this unique and threatened system of coral reef is already close to going into red. Certainly, this is a color code. We are using it based on our assessment. So there is a clear role for ecosystems to guide the setting of long-term global goals relative to pre-industrial, considering levels of risks for vulnerable systems. But what role for experimental biology? Let's take this a little further. The risk actually is defined considering the vulnerability of that system, and this is where experimental biology comes into play to characterize that exposure and the climate hazard. Possibly also the adaptation measure taken by humankind can be a challenge on species, or the mitigation measure taken, because all of these will be large-scale changes to systems. Risk is then connected to projected impacts. Impacts influence socioeconomic processes. They give us information on what kind of emission and land use change would be important in this context, and there are feedbacks to climate. So it's a characteristic, it's an element in a complex system. There are feedbacks, there are opportunities. Russia likes the expansion of the agricultural area that they are seeing. Some of this may be true for Canada as well. But there's a price to be paid at lower latitudes in that context. The impacts that are ahead of us provide motivation to take action. So the risk assessment is an important aspect in setting climate targets through risk comparisons. And we do this for different time windows, and this is just a schematic showing the level of risk, and it's also showing how much we are able to reduce the risk by human adaptation. At a species level, we would say this is telling us how much we can reduce risks through evolutionary adaptation. But we are already seeing from what's happening that adaptation rate is rather slow in relation to the velocity of climate change. And the capacity to adapt may become narrow as we go into high-degree temperature changes in a climate future. And the near-term 2030s to 40s will be our 1.5-degree world. And the question is whether we can keep temperatures at that level. Certainly, there is a potential for mitigation to reduce risk, and that is not included here. But now the modeling experiments come into place. Building on basic biological insight, what can we do and how can we do large-scale projections into the future that provide a strong message to policymakers that there may be something ahead of us, a future ahead of us that we do not want. Now, this is a projection taking into account temperature window data for 1,000 species of fish and invertebrates, something you cannot do by just using Excel files. So there is a mathematical exercise in relation to temperature changes projected. We see there is a loss of biodiversity projected in the lower latitudes. The bluish areas indicate where we may have some species gain by 2050 to 2060, we may actually have some gain in fisheries catch potential as we are currently seeing in the Barents Sea. This is simplified, but it provides a global look into the challenges ahead of us, and we specifically talk about projections and not about predictions. There is a clear difference between the two in IPCC terminology. If we do our risk assessment, we see that the risk is increasing. There will be some scope for adaptation, especially for industrial fishing fleets. They can follow the fish stocks, but the local artisanal fisheries will be challenged. They will not be able to adapt, and they will lose a large fraction of the fresh food that is available to them still currently. Another driver of change is ocean acidification. This is how far we could take the analysis in 2014, up to 2014. This is giving you an overall picture about the degree of ocean acidification by end of century. As a global picture, you see the ecosystems affected, the warm water corals, the cold water corals, the purple color, warm water are green, and then the yellowish areas are the ones where humans collect calcifiers for their consumption. So essentially, it's showing the drivers and the systems affected, but no projection. We have a meta-analysis and had it available at the time where we looked at the CO2 sensitivity of relevant organism groups. The fraction of affected species in a group at a certain CO2 level will indicate us about the vulnerability of the phylum. And you see the vulnerability is different. Crustaceans are less sensitive than mollusks. Cold water corals are less sensitive than warm water corals. But now I can see one point, this driver doesn't operate alone. We have already talked about temperature. So they are interacting. What we knew at the time, effects are maybe exacerbated by warming extremes, but we need to say generalized mechanism-based spatial modeling approaches still need to be developed for this challenge. The SEB mafia working on fish would ask, where are the fish? And rightly so. So this is the full meta-analysis as we did it, and there is fish included. But at the time, there were these messages coming up that fish behavior is disturbed. And the fraction of species disturbed in that respect is relatively high, indicating a high vulnerability of the group. But we do not have additional lines of evidence telling us that this group is vulnerable long-term, that they do not have a possibility to adapt to these challenges, which we can simulate long-term in our laboratory, but the real long-term would be beyond our lifespan. So there is no fossil record indicating vulnerability to ocean acidification long-term in fish. And this is why these findings did not make it into the summary for policymakers, because the confidence was assessed to be too low. We've made some progress in that respect, and this comes back to the differences between life stages and their different tolerance ranges here shown with respect to temperature again. If we look at this at the egg stage, embryonic stage for Atlantic cod, we see we get a certain temperature window. It's relatively narrow, and we see that this temperature window shrinks under the impact of ocean acidification as projected by the end of century under business as usual scenarios. Now we are now collaborating with our ocean physicists and climate physicists and putting this into projection maps. Comparing unabated warming with reduced warming depending on emission scenarios and the Paris Agreement, 1.5 degree warming as a goal. What we see is actually that under all of these, we see a certain reduction in egg survival, telling us about spawning habitat suitability for the species. But what we also see is that this effect is small under 1.5 degree warming smaller than under the other challenges, and this may maintain current spawning habitats. We do not really know whether this species would actually go much further north and shift its spawning grounds according to environmental temperature, because there may be other challenges ahead. So cod and other species are at risk of losing major spawning habitats if climate change continues unabated. That's a relatively safe statement, and limiting global warming to 1.5 degrees will minimize habitat loss. So this is the kind of projection we need to be able to tell to the policymakers. From a more global view, ocean acidification comes with increased risk. In the near term, with a moderate degree of acidification, there may be some adaptability, again for human fisheries activities, because we may be switching to exploiting more resilient species, and we may be able to reduce other stresses so that they can be more tolerant to ocean acidification. But if we let it go unabated, this capacity to adapt will go away. Now let's look at the interaction between the other two challenges, warming and oxygen loss. If we take our projections based on routine metabolic scope further to a more general information and look at areas as especially the North Pacific, but other places as well, we see that our critical metabolic index suggests a constraint on thermal habitat by 20% overall. And if we go to places where we have already oxygen deficiency, this may be a 40% effect. These are projections that are in the literature, need to be assessed in the 6th assessment cycle by the respective author teams. But they tell us about the challenges ahead and how much it is important to look at the interaction between climate drivers. And this may also cause relocation of fishing activities. Specific fisheries like the one for Humboldt squid may benefit, as this is hypoxia dependent, and controlling nutrient input and pollution may also be important. But there is a pretty steady increase in risk depending on the degree on climate change, and capacity to adapt is only in the near term. So we then made more efforts to bring the risk assessment together for the different ocean sectors and for the organism groups. And you see how this has been done, going from seagrass to mangroves to warm water corals to pteropods, bivalves, krill, finfish, and where these risk thresholds are in relation to different climate futures, with a very low threshold for warm water corals, even touched upon in the present day. And then we have the services based on these sectors: carbon uptake, coastal protection, recreational services, fisheries, finfish fisheries, and so forth, and their respective risk thresholds. Informative, but I have an even more powerful picture next. But what this tells you is that capturing the diversity of organisms, ecosystems, and ecosystem services in a coherent framing makes sense, because it makes risk comparable and helps to formulate strong messages to policy. So this is again a burning ember approach, somewhat different, building on the data of the last slide, but tying it together for different climate futures. And the black line characterizes the recent 0.8 degree climate change compared to pre-industrial, where for all sectors we are in the yellow range. Whereas if we go to a 1.5 future, which is the solid white line, it still keeps us pretty much in the yellow range, but it goes into the red for the corals, and that's unavoidable. If we go and consider the current ambitions of the nations to do something about emissions and emission reductions in their so-called INDCs, the intended nationally determined contributions, then these are the dashed lines. And depending on the calculation and the projection and the climate, the response of the climate system, the climate sensitivity, the position is a little different, but all of them take us into a red area and purple area. So high risk and very high risk for many of these sectors. telling us it makes sense to go for certain climate targets, telling us it makes sense to go into ambitious mitigation, reduction for conservation purposes concerning biodiversity and the contributions of nature to people as ecosystem services are currently framed in the IPBES and the IPCC. So what role for experimental biology in climate-related assessment reports? Let me summarize. Mechanism-based science is important for the detection of climate-related phenomena and their attribution to climate drivers because they enhance confidence. They are needs to form the basis of improved and more precise modeling. So we need to do better with respect to our projections until we may finally then come into when we can talk about predictions. Mechanism-based science needs to connect across disciplines and contribute to an integrative view, building and drawing on multiple lines of evidence. It can provide basic science messages relevant to policy, and they can be lifted to the summaries for policymakers, as you have seen in the examples listed. But we should not forget mechanism-based science is also useful to enhance and exploit the science basis of solution options, addressing the capacity and the limits of adaptation, identifying the clear limits, rather than giving policymakers the message, oh, all living systems on the planet can adapt to climate change. Identifying ways to enhance blue and green carbon storage to restoration, mechanisms, supporting sustainable development, conservation, protected areas. So bringing diverse phenomena together into a coherent storyline would strengthen a role for experimental biology in climate-related assessment reports. Now let me close with this one. The Paris Agreement provides a sense of urgency, but some people just don't want to know. So there is inertia in society. We need to overcome societal inertia and inaction in transformation, because transformation will be far-reaching. It will be affecting the lifestyle of all of you over the decades to come. What we can say based on our previous efforts is that ocean findings are among the clearest on climate impacts in relation to the degree of climate change. Ocean findings help strengthening the UNFCCC process and global stocktake. And ocean findings are needed to enhance and exploit the science basis of solution options, and experimental biology findings have played a role in enhancing the confidence in that context. So experimental biologists should join forces and develop strong consensus statements and key messages from their field to make a contribution to the discussion in the policy arena, and that needs to be happening very soon. We are currently doing our best in the 6th assessment cycle to bring all of the detailed information concerning climate change impacts, mitigation, and others together. And just to show you how we are doing this, we are currently finalizing the special report on global warming of 1.5 degrees to be approved in October. That should be fed into the facilitative dialogue of the UNFCCC and the next COP. And they are currently working on what governments should do about climate change in light of the Paris Agreement. So this will be sending strong messages there. Then we are having special reports in the pipeline, one on oceans and cryosphere, one on land under climate change, to be approved by the end of next year. There's a parallel effort on doing emission inventories on countries. And then there are the main reports with their approval dates for Working Group 1 in April 21, for Working Group 2 in October 21, Working Group 3 in July 21. So it's all staggered and well planned, very tight schedule until then. And in April 2022, we will bring the efforts of the 3 working groups together in the synthesis report and the first global stocktake by UNFCCC, telling us where we are with respect to emission reduction efforts and where we should be will be happening in 2023. So it's not going to be boring for us in the years ahead, and it would be nice to see more input by experimental biology in these efforts. All authors, review editors selected based on government and bureau nominations. That's done. So joining one of these reports as an author, as a lead author, would only be possible in exceptional circumstances. But you can still make a contribution, become involved as expert reviewers, pushing the authors to say something about cause and effect mechanisms. Maybe you can be contributing author to these reports, filling in specific information. You can send this in to the author teams as the reports are developing. And the role of these expert reviews should not be underestimated. The special report on global warming of 1.5 degrees, due to the high level of interest, got 25,000 comments. And all of these comments need to be answered by the author teams. So they cannot just ignore comments. They cannot decide to do that like you would do that if you get a bad review on a paper. So with that, I would like to close. I would like to stimulate you to be interested, to become involved, and to watch this process more closely and take opportunity as it comes along. Thank you very much for your attention.